We request all the participants to be seated for the next session. We will start immediately. I will start. Shall I start? Yeah. Welcome back to the second technical session of Agriculture and Food Processing. Let's start with the first speaker of this session. The first speaker is Menshchikov Ilya Evgenyevich. He is Deputy Head of Department at LLC Unisob, Russia. The title of his talk is Atmospheric Absorption Water Harvesting Hydropanel. He will be joining online, so please switch on your video.
Thank you very much for kind introduction. Today I would like to discuss with you a problem of access to pure water and alternative base and the methods for its harvesting. And my presentation is dedicated to our promising invention, atmospheric absorption water harvesting hydropanel. Everybody knows that access to water with high quality in sufficient amounts determines the quality of people's lives. According to United Nations and UNICEF statistics, more than 2.2 billion of people suffer from lack of water, and 700 million of them could be displaced by intense water scarcity by 2030. Here you can see that a lot of countries from Central Asia, Middle East, Africa, and America suffer from lack of water. Moreover, about 70% of SCO-associated countries located in the Middle East and Asia are likely will face the risk of extremely high water stress by 2040. Russian Federation possesses enormous deposits of fresh water, but even in this country, more than 20 million people face problems of access to drinking water right now. Cottages, gardeners, and owners of private houses. Of course, we can mention well-known methods for water supply, such as artesian or bottled water. However, they don't meet modern ecological and socio-economic requirements. And what about generation of water from atmospheric air? We think that this method has a chance to become a new reality in obtaining pure drinking water. Today, we already have technological solutions for water harvesting from the air, for example by condensation using refrigeration machines. However, this method requires conditions of high air humidity and have decreased energy efficiency due to usage of electricity or hydrocarbon fuels. Autonomous hydropanels based on absorption principle is a new solution which can turn over the market of water supply in the nearest future. Operation of our device is based on the utilization of a special hydroscopic absorbent with high activity to water molecules. Cyclic work of a hydropanel includes two modes, day and night. At night, moist air enters the hydropanel through the filter. Moisture from the air is absorbed by a layer of special absorbent. Dehumidified air returns to the atmosphere, and the panel, 1 square meter of the hydropanel, can accumulate up to 5 liters of atmospheric moisture per night. In the daytime, solar energy heats the hydropanel and the absorbent layer. Due to heating, absorbed water is released from the absorbent in the form of water vapors, which condense on a special surface and transported to storage tank. Then clean water from the storage tank is pumped through the mineralizer and sent to the consumer. This is our device, a solar-powered generator of pure drinking water from atmospheric air. On this slide, you can see our second prototype of the device. The idea of using hydropanels is that they can be fixed, for example, on the roof and connected with the water supply system of the building or a house. Here you can see the list of advantages of the hydropanels. The system provides water of high quality without contaminants. I also have to highlight such advantages as absolute environmental friendliness and complete autonomy. Unfortunately, the main disadvantage of the system is dependence on weather conditions. You will see it on the next slide. On this slide, you can see the dependencies of daily performance of 1 square meter of absorption hydropanel on relative humidity of ambient air at different weather conditions, such as clear sky, cloudy, and mainly cloudy weather. It is obvious that the highest performance of the device can be reached on sunny weather due to maximal solar heating of the absorbent layer. Also, water harvesting efficiency increases with relative humidity of ambient air. However, it doesn't mean that utilization of the panels in the regions with cloudy weather is impractical. Special technical solutions can be employed, such as additional heaters for absorbent layer, specially prepared absorbent with tailored porous structure for selected working conditions, and so on. Our main competitor today is source water generator produced by Zero Mass from the United States, which employs absorption principle for water harvesting from the atmospheric air. In comparison with the source Zero Mass, our device has such advantages as 3 times higher water harvesting capacity from 1 square meter of the panel, which reaches 5 liters per day, lower cost of the device, and as a result, lower cost of 1 liter of water for the final consumer. And also extremely low humidity threshold of 5%. For example, source panel or condensation machine operate at humidities higher than 10 and 60% respectively. It is also expected that various consumer groups will be interested in using our hydropanels, such as travelers, farmers, private households, small businesses, non-profit foundations, and charity organizations. It is expected that overall estimated volume of B2C market of the hydropanels can exceed $9 billion of US dollars for Eurasia, Africa, Australia, and the United States. If we consider B2B market, such segments as prefabricated buildings, motorhomes, and glampings can be promising consuming areas of our technology. By this time, we have produced 2 prototypes of the hydropanels and successfully fulfilled tests in Moscow and Krasnodar region in Russia, which prove the concept and main parameters of the device. We also participated in several business acceleration programs and became a member of Skolkovo Innovation Cluster. MVP of the panel for domestic use and start of its production is expected this year, 2023. We are also working on the protection of our intellectual property. We have already received Russian invention and trademark patents as well as utility model patent, and now we are waiting for the validation of several overseas patents. Next steps of our development will include mobile solution for water harvesting, which is under high demand for temporary settlements, hospitals, and others. High-capacity water farms for industrial applications, for example, agriculture, and development of special line of water to individual tastes and properties. And this is our team, which consists of experienced high-class scientists and engineers. And thank you for your attention.
Thank you, Dr. Evgenyevich, for such a nice presentation. Our next speaker is Chaomin Yin, who be Academy of Agricultural Sciences, China, and the title of his talk is Isolation, Characterization, and Bioactive Evaluation of Polysaccharides from Edible or Medicinal Fungus. He will be joining online. And questions will be taken at the end of the session. Thank you.
Dear colleagues, ladies and gentlemen, I'm Chaomin Yin, a researcher from Institute of Agro-Products Processing and Agricultural Technology, Hubei Academy of Agricultural Science of China. It's my honor to give a talk. in the second Young Scientists Conference of Shanghai Cooperation Organization. The title of my talk is Isolation, Characterization, and Bioactive Evaluation of Polysaccharides from Edible or Medicinal Fungi. The content includes 4 parts. The first is personal profile. Now I'm working in the Institute of Agro-Products Processing and Agricultural Technology, Hubei Academy of Agricultural Science of China. My research mainly focuses on the biosynthesis, regulation, green and efficient isolation, and bioactive evaluation of bioactive compounds from edible or medicinal fungi. The second part is research background. In 2021, the total output of edible fungi was 41.33 million tons, and the total output value was 347.56 billion yuan in China. Currently, more than 75% edible fungi in the world were produced by China. This shows that the edible fungi industry is a large industry in China. Because of the coronavirus pandemic, the global economic downturn seriously, which blocked the edible fungi export. On the other hand, edible fungi are not a necessary consumer products like rice, flour, or meat. Thus, the sales of edible fungi are limited. One word to describe the situation. Edible fungus price fell, growth did not earn money, and edible fungi enterprise lost money. Another perspective. Most of the edible fungi processing in China are primary processing, such as drying and simple packaging. The ratio of deep processing less than 10%. The deep processing includes drugs, cosmetics, health products, and so on. As everyone knows, all these products are involving the bioactive compounds. However, the health mechanism of bioactive compounds in edible fungi are not clear. Green and efficient preparation technology, as well as the directional precision processing design, are lack of. Therefore, we concerned the mushroom bioactive compounds. Our research idea was as follows. Firstly, established extraction method, then study the health mechanism of bioactive compounds, and developed the key processing technology. Finally, carry out the health product design. The third part is the main research contents and highlights. Firstly, we established the enzymatic hydrolysis microwave ultrasonic method for mushroom polysaccharides isolation, and the extraction rate improved 50% when compared to the hot water extraction. We developed a polymer complexing precipitation method for mushroom polysaccharides. The purity of polysaccharides obtained was higher than 70%. Accordingly, we obtained the authorized invention pattern of China. We found the gamma irradiation treatment could significantly increase the purity of polysaccharides, reduce its molecular weight, and increase its solubility. The changed characteristics increased the application of polysaccharides. We analyzed the fine structure of cytophyllum chromium polysaccharides and found the SCP1 is an alpha-glucan that contained mannan in its backbone. SCP-1 can effectively inhibit the growth of lung cancer cells, and the cells of toxic-induced effects may feel PEX or PI3K signaling pathways. We found the gamma-irradiated cytophyllum chromium polysaccharides ISFP can reduce the weight gain of mice and increase the relative content of beneficial bacteria in the intestine of mice. In human gut microbiota fermentation experiment in vitro, we found the ISFP greatly increased the relative content of tannalaceae, which is an important way to lose related gut microbiota in human intestinal tract. Moreover, we found the polysaccharides from Sanhuang porous family can enhance the immunity of mice by improving the release of cytokines, immune globulins, and the lysosome activity, increasing the level of beneficial gut microbiota, especially Bacteriophages and Lactobacilli, as well as the content of SCFAs. Highlights: we established the green and efficient isolation technology of mushroom polysaccharides and analyzed their health mechanisms. Based on above, we obtained the third-party scientific and technological achievements evaluation. We developed the products of polytablet and commercialized the scientific and research achievements in the enterprise. The last part is the acknowledgement. Our research is supported by Hubei Academy of Agricultural Science, National Natural Science Foundation of China, Department of Science and Technology of Hubei Province. Thanks for your listening.
Thanks, Dr. Chaomin, for your nice presentation. Our next speaker is Suldina Ekaterina Vladimirovna, who is a faculty at Federal State Budgetary Educational Institute of Higher Education, Ulanovsk State Agrarian University, Russia. And the title of her talk is Bacteriophages for Agriculture and Food Industry.
Bacteriophages for agriculture and food industry. For the first time, the possibility of using bacteriophages in the prevention and treatment of infectious disease was demonstrated in 1917 when sensational news spread among microbiologists that Felix de Havel had discovered viruses that are bacteria, and on their basis he managed to develop drugs for the treatment of soldiers infected with dysentery. Bacteriophages are viruses characterized by a specific ability to selectively infect bacterial cell, followed by lysis of the host cell or the formation of lysogens. For more than 80 years, in the former Soviet Union and later in the Russian Federation, the branches of NPO microgene have been production more than 19 variants of drugs containing bacteriophage to the most common pathogens of human bacterial infection. For a long time, diagnostic test systems based on phages have been produced in our country, allowing to identify pathogenic microorganisms that cause listeriosis, anthrax, plague, cholera, etc. The staff of the Department of Microbiology, Virology, Epizootiology, and Veterinary and Sanitary Expertise of the Ulyanov State Agricultural University has been studying bacteriophages for about 40 years. The choice of this direction is due to the fact that in recent years the idea of the etiological role of preventionally poorly studied bacteria and the pathology of animals and humans, which are widespread in nature, has expanded. Most of them are often part of the normal microflora of gastrointestinal tract of animals and humans, and also inhabit object of the external environment. During this time, a specific and technical base and a significant research team were formed. More than 35 candidates and doctors of science were treated. Currently, with the help of bacteriophages, it is possible to solve a significant number of problems in the field of agriculture and the food industry. Bacteriophage-mediated control, phage bioprocessing, prophylactic, fungi identification and indication, disinfection of facilities, diagnostic, treatment, and prevention of disease. Our team is actively working in all their areas. A lot of work is carried out to isolate phages active against many pathogenic bacteria. We study biocontrol properties of isolated phages, select phage isolators with characteristic properties specific for use. For the first time in the Russian Federation, effective means of bacteriophage-mediated biocontrol for a number of microorganisms have been constructed on the basis of isolate from environmental objects and characterized bacteriophages. A multi-stage team of control and testing measures of a new category of bioconservation and technological processing based on bacteriophages has been formed and adapted. The pilot technology for the production of preventively phage cocktails for prophylactic use has been improved. The effectiveness of the use of phage for the identification and indication for bacterial infections agents has been proven. The elevator for the safety and effectiveness of phage as biodisinfectants has been created. That is for me. Thank you for your attention.
Thank you very much, Dr. Suldina, for such an interesting presentation. Our next speaker is Peixi Zhu, Zhejiang University of Technology, China, and the title of his talk is Selective Extraction of Active Components from Nature Products. If you are online, please switch on your video.
Hello everyone. Today my topic is selective extraction of active components from natural products, and I will cover these three parts. Natural resources contain a variety of materials, including essential oil, flavonoids, and other components. The utilization of the natural resources should keep in mind that resource conservation and environmental friendliness is the most important thing. At present, the extraction of valuable substance in the industry mainly depends on traditional extraction methods. Although they are very simple, the extraction efficiency is very poor, and they take long time and have high temperature and they use very high solvent consumption. Thus, mechanical chemical assisted extraction, or MCAE, is developed to do the extraction. And mechanical chemistry is the process by which mechanical forces act on solid objects, and the mechanical energy is converted into substance and internal energy to reduce the changes. The mechanism is still under investigation. However, there are two important factors affect the efficiency, and this process is the cell wall fragmentation process. And as most bioactive components exist in their cell walls, the cellular components block the release. But the mechanical chemistry process can broken the cell wall completely, and it has already been observed by SDM technology. And second, under the action of high energy by breeding process, the target compound could react with chemical accessories and transfer into water-soluble components, and as a result, MCAE could use water as the extraction media. So in summary, MCAE technology has a characteristic of eco-friendly and receiving water solubility and high extraction yield, and selective and simple. And this is a recent work done by us. The citrus, in Chinese we call Dai. It's a common grown in Zhejiang and Fujian province in China. It's very interesting because if the red fruit remain unharvested on the plant and kept on the tree until the next summer, they could become turn back to green like fresh fruits. So they call Dai. In the study, we use MCAE technology to extract the essential oil flavonoids and polysaccharides. The extraction process for the essential oil are as follows. The fruit, as flower, was dried and pulverized into small size, and then mixed with select solid region and graining for several minutes. And after that, mix materials with solvent for several minutes and filtered. And after removing the solvent, the essential oil could be obtained. And the conditions for the MCAE technology was screened to get the final condition. And using this method, the process could be done in room temperature with extraction yield far better than the two other traditional methods. And it could be observed from SEM data that when it was treated by mechanical reaction, the particle size is smaller and the complete shadow, thus it could fully release the essential oil we want to extract. We also develop methods for extraction of flavonoids and polysaccharides. And up to now, we have developed a series of methods for the extraction of various types of compounds from natural resources and applied many patents. And these are the plants we already realized in industrialization by us. And the technology solved the problem of the efficiency, high pollution, and other problems they met, and they have good production. In complementary to the following studies, as we noticed that the plant could have property of multi-components and multi-targets in order to fully find components for a particular drug target activity and know more about quality of the extract. We use enzyme immobilized nanoparticle to screen the active components for a particular target. The target was immobilized with a special enzyme or protein, and after that it was incubated with complex extract of the plants and separated by magnetic. And after denaturing of the nanoparticle, it could release the active components, and we could know the active the active component. And further, by using LTMS, we could know the structure. So in summary, based on the current work, we will continue to explore MCAE to select extract components from natural resources. And in order to gain more results for the mineralization, research related to the quality of the extract also our interest. And this is my team is leading by Professor Wikutu, who is the director of Collective Innovative Center of Yangtze River Delta Region Green Thematics. We also had the pilot scale lab for the application of MCAE. We also had analytical lab for support of our research, and I'm in charge of the platform. And we have our joint lab for mechanical chemistry with Russia, and we have a relationship with companies to help them solve the problem and do the industrialization. Finally, this is our campus. We have 3 campuses, and we are in the first picture, the Morgan campus. And welcome to our school, and welcome cooperation with us. Thank you.
Thank you very much, Dr. Peixi, for very nice presentation. It's my kind request to all the participants and volunteers to maintain silence during the talks. Our next speaker is Yengkhom Singh, who is an assistant professor at Central Agricultural University, College of Horticulture and Forestry, Arunachal Pradesh, India. And title of his talk is Curcumin-based nanoformulation for shelf life extension of horticultural crops. Can you please raise your hand so that our camera can capture you?
So, friends, a very good afternoon and warm welcome to the second SEO Young Scientist Conclave. This is Dr. Yengkhom Singh from Central Agricultural University, College of Horticultural and Forestry, Pasighat, Arunachal Pradesh, India. Today, I will be talking about the curcumin-based nanoformulations for shelf-life extension of horticultural crops. Before begin my talk, let me brief about my university. So our university, the Central Agricultural University, Imphal, is one of the premier institutions working on the agriculture sector. I would say it's the first centrally funded agricultural university in India. We have 13 campuses across the northeast India, and among them, the College of Horticultural and Forestry Pasighat is one of the premier institutions working on the horticulture and forestry. So, coming to the edible coating, as we know that the edible coatings are the coating, basically the thin films, which are usually used for preservation of the food over coating the fruits and vegetables. They have the properties which makes them, weighed against the moisture, gases and solid movement. And of course, the edible coatings are eco-friendly, highly safe, and low price, which makes them the promising, the packaging materials, food packaging material. Now coming to the turmeric, as we know, the turmeric, the Curcuma longa, is an ancient herbaceous perennial spice belongs to the family Gingiveraceae. It is native to the Southeast Asian countries, including India, China, Indonesia, Thailand, and some parts of the Africa and America. In India, it is extensively available in Andhra Pradesh, Tamil Nadu, Odisha, etc., and some northeastern parts of the India. It is proud to say that the Indians are the largest producer and exporter of turmeric across the globe. We produce around 80% productions and an exporting of 45%. And the turmeric are being used as a spice in the culinary items, in the food preparations, in the coloring agents, cosmetic and pharmaceutical, the drug preparation, etc. So this is the photograph that we have taken from the Riga village, the Siang district, Arunachal Pradesh, which is very, very far away from the mainland India, from where we have visited all the team. You can see the farmers field, okay, the villages. And this, the Riga village, is the place where the scientific interventions are very far away and nobody has, explored such kind of places. So we went for the sample collections. You can see the collections of the samples, the different varieties of the Curcuma longa collected from across the northern state. And we analyze the morphological characters. You can see the different colors, different colors ranging from yellow colors to the dark reddish colors to the white color, etc. And of course, the darkish yellowish color, this one. So they have, more curcumin concentrations as you compare with other light yellow colors. So the curcumin I was talking about, the curcumin is the main constituent, the bioactive compounds present in the turmeric. And it's not only the curcumin which are found in the turmeric, the other plants, the Curcuma aromatica, Curcuma faecalis, the Curcuma gyroideae, they also seems to have the curcumin, but in a very less. As already I talked, the Indians are the largest producers of the curcuma across the globe. And when we talk about the Indian state, the Andhra Pradesh is the number one, and then the Tamil Nadu and rest of the other state of the. So if you look to the northern state, the northern states are also producing the turmeric in a large quantities. So among the northern state, the Meghalaya, Sikkim, Mizoram, they are leading ahead, and some of the Arunachal Pradesh and Manipur, Tripura, etc. So now, if you look to the trends of productions of the Curcuma longa, starting from 1990 to 2019, the trend is increasing. It is inclining, This indicates that a large number of turmeric are being consumed across the globe. So this diagram, it is indicating. So we have also, the characterized They are the spices found in the northern state. And very interestingly, the turmeric seems to be having zero, the quantity of the cholesterol you can find. See, so that is the idea, that is the thing where the turmeric can be, utilized for the culinary purposes and some other, the drug and pharmaceutical companies. So, the cholesterol deposition is causing a lot of problems in the human being. So, talking about curcumin, the curcumin that we extracted from the rhizome of the Curcuma longa, these are not in a pure form. These are the ternary mixes of the curcuminoids. So the curcuminoids usually contains the curcumin, the DMC and BDMC. So these are all in contents in a mixer form when we extract from the rhizome of the Curcuma longa. So curcumin may have different structures like the curcumin 1, 2, 3, and then the hydrocurcumin, the cyclohydro, the cyclocurcumin, etc. So talking about the applications of the curcumin in biological aspect, we have antioxidant property, anti-inflammatory, the anti-cancer, the antimicrobial, etc. So these are all, the mode of actions which I have mentioned for the curcumin in the human applications. Now, being as the agricultural aspect, we must understand the calendar of operations of curcuma longa. The right time for sowing in India is in between the May and June, and the harvesting time starts from the January. So when the plants sown and it reaches to 9 months, then you can start harvesting this curcuma longa, and it is the perfect time to harvest starting from January to March. So the post-harvest processings of the curcuma longa, one thing that we are missing, that is the the treatment, the pre-treatment of the rhizome, the curcuma longa. So it is very compulsory and very important to treat with the 1% sodium bicarbonate. So if you do so, then the fungal infections down the line where you keep for preservations, it will be reduces. So it is need of the farmers are usually not doing this treatment. Now, the value-added productions from the curcuma longa, many lot value-added productions can be, get from the curcuma longa, but our main target is the curcumin as the edible coating. So, the curcumin as the edible coating, that's the interest that we will be looking about. When we say the edible films and edible coating, a large number of, the materials, you can find it starting from the proteins, the polysaccharides. So our main focus is the curcumin based nanocomposite. Why we have taken the curcumin as the edible coating? As already I mentioned, the curcumin are having the antioxidant, the antimicrobial, and these are non-toxic to the environment as well as to the human beings. But one should have the knowledge of the doses of curcumin in the food. The recommended doses is 20 to 500 mg per kilo in a day. So it should be, we should understand. Now, the edible coating. So the edible coating, when we apply on any kind of fruits and vegetables, so it actually makes a barrier upon the membrane, and thereby it protects the infections, the fungal infections, the microbial infections to degrade the fruits and vegetables. So a different class of materials like polysaccharides, lipid, protein, these are all used, extensively used, and you can find in the literature also. But the composite has more, impact in making the barrier, because that's the barrier, okay, the good, that's the thing that we are actually looking for. But rest of the other materials will have the pore mechanical and the barrier property. So that's why the composite has more importance as compared to materials. So we make the edible film based on alginate, the turmeric in 1:13 ratio. So these are all the characteristics of the metals that we have developed. Now the composite, the curcumin, alginate composite, it was confirmed, by the infrared spectra, where you can see the two colors in the bowl. The yellow one is the composite, and another one is without the composite. So this is how the we are actually applying on the fruits and vegetables. So this diagram explain how we are the nano curcumin can be actually used as an edible. So there are usually a not many methods, but usually three methods we are applying in the coating, dipping, spraying, and brushing. So we found in all the dipping, spraying, and brushing method, the curcumin nanoformulation is showing, the shelf-life extensions of the fruit vegetables. Apart from that, very interestingly, when we increase the nanocurcumin concentrations, the antioxidant property is also increasing. So that's how this figure is explaining. Apart from that, we also synthesize the green copper nanoparticles from the curcuma longa. This is how we have characterized the copper nanoparticles. So these are all the histograph and the micrograph of the FECM images and the XRD images for the copper nanoparticles that we have synthesized. And by looking through the literature, we found that these nanocurcumin are these are biocompatible, these are edible, coating, which it can affect the every parts of the body, but without hampering the body. So these are all my publications from this nanocurcumin based formulations in some of the the these are all my collaborators working together, ranging from the DBT Center of Excellence, the NERIS, the NIFTEM, the Tezpur University, the IIT Guwahati, the NIS Jorhat, CSI Laboratory, CIT, Kolkata, etc. So lastly, I give my sincere acknowledgement to the Department of Biotechnology for funding my entire research work, the Unat Bharat Abhigyan, which is the IIT Delhi flagship program, and also the Department of Science and Technology for funding my entire program. So lastly, I'm very much thankful to the organizers for giving me opportunity to explain my research work in front of the Asia nations. So I'm very much, I'll be happy to uh, get some queries from the audience, as well as I'm looking forward for having the collaborations. So once again, thank you all. Thank you so much.
Thank you, Dr. Singh, for such a nice presentation. Our next speaker for this session is Zanar Kadasheva. She is head of the chair of Chemistry and Chemical Technology at Khalil Doshmukhimodo Atrio University. The title of her talk is Development of Irrigated Cultivated Grasslands on the Salinated Lands of the Caspian Region. Ma'am, can you please show your hand? Dr. Zana, can you raise your hand? Yeah, thank you.
Dear colleagues, let me introduce myself. My name is Shana Arkadashova, and I am head of the chair of Chemistry and Chemical Technology, Khalil Doshmukhimodov Atrio University. The topic of my presentation is development of irrigated cultivated grassland on the salinated lands of the Caspian region. Mentioned conditions of the Caspian lowland are adverse to the cultivation of forage crops and the high demand for milk as fresh dietary and imperishable food in the region, especially especially for children, dictates the need to reclaim sudden and suitable plants. A high rate of soil salinity because of the close near surface location from a soil depth of 50 to 70 centimeter of the so-called maximum salt accumulation horizon contained salt to a very high degree of 1.8 to 2.3% in the number of toxic ions stated to be the limited factor for planting and development of grasses in this region. The high concentration of salt prevents root penetration of the cultivated plants on this horizon, except for millet. As a result, cultivated grasses suffer both a lack of nutrient ingestible from the deeper soil horizon, 50 to 200 centimeter, and the toxic effects of salt that cause the suppressed growth and development of the above ground mass. All these affects the productivity of forage plants, which in the indicator determine the choice of a particular plants in a given region. Such circumstances defeats crop selection with a certain degree of salt resistance, economically justified and environmentally sound productivity on short profile, so up to a depth of 50 to 70 centimeter, where the root system can develop. All these determines the relevance of the study. 2016 and 2018 were undertaken on meadow brown soil, and in 2021 available meadow soil in the maximum salt accumulation horizon of latter was located at 50 centimeter depth. The selected harvest composition was unchanged and included alpha-alpha, orchard class, Russian guild trimeller, and it is herbal mixtures. According to the chronometric composition, the soil very medium and heavy loamy with a 0.8 to 1.2% humus content, 0.15 to 1.1% of total nitrogen, and phosphorus and potassium content were 1.3% and 1.75% respectively. So I provide these mobile forms of nitrogen 4.5 milligram per 100 gram of soil, and a weak degree phosphorus 1.4 milligram per 100 grams of soil, and a high degree potassium 38 milligram per 100 gram of soil. The soil are falling to a weak medium and high degree, whereas the content of toxic ions in 0.3%, 0.6%, and 0.9% respectively. The type of salination is fluoride sulfide. The variance is present in tables where alpha-alpha was the control in the experiments. The data on the green mass of perennial grasses studied in the experiment, the ground water level in the area and the study lays lower than 2.4 to 3.4 meters, and it does not affect the growth and development of productivity of the study's crop. The penetration path of the studied crops on all type of soil in the Astral region in a 50 to 70 centimeter layer. There is a maximum salt accumulation horizon with a high contact of toxic ions more than 2%, where the root system of arid crops do not develop except for millet. In table horseshoes, the pair combination needs 6 moving throughout the growing season, similar to the partial mass crop of the grass mixture grows from spring and early summer to mid-July, peaks in the second half of July and first part of August, and then declines to levels similar to those of early summer. Notably, in accordance with the specification of the developed experiment techniques, the grasses were removed to pastel moss and the height of 25 to 35 centimeter, primarily during the phase of the entering tube. Grass mixtures with the fortification of sweet millet turned out to be more productive compared to grass mixture, in which alpha-alpha was included. Grass mixtures of millet, orchard grass, and the brown grass produced the highest yield as seed for combined mixtures with wheel drive, and also the three-part combination of orchard grass, brown grass, and alfalfa proved to be more productive compared to the combination of orchard grass, alfalfa, and wheel drive. Balance of soil concentration is maintained in top 0 to 70 centimeters soil layer. High productive salt resistant crop can be grown on medium saline soil. Long-term experiments showed effective cultivation on the large number of crop grown on saline irrigated lands. Millet as well as its mixture with smooth brown and pork food showed high salt resistance and productivity, providing grain mass yield. The research was carried out within the framework of the project entitled Development of the New Technology for Restoration and Rational Use of Pasture research funded by the Ministry of Agriculture of the Republic of Kazakhstan. For your attention.
Thank you very much, Dr. Zanar, for such an interesting presentation. Our last speaker for this session is Tripti Singhal, who is a research associate at ICAR, Indian Agricultural Research Institute, New Delhi, India. And the title of her talk is QTL Mapping for Grain Iron and Zinc Content in Pearl Millet. Can you switch on your video, please? Dr. Tripti?
Hello everyone. Thank you so much for giving me this opportunity to present my work in second Assyrian Scientist Conclave. This is Dr. Tripti Singhal from ICAR, Indian Agricultural Research Institute, India. My research topic is QTL mapping for grain, iron and zinc content in pulse millet. The food security challenge is successfully achieved in many of the developing countries. It is not only enough to generate more food in form of staple crops, but also appropriate nutrition is required for everyone to end hunger and malnutrition. Now, more than half of the global population are affected by its harmful consequences. However, India alone shares one-third part of it. Among all the trace elements, quite a few trace elements such as the iron and zinc are very important for proper human health. The deficiencies of these micronutrients affect more than 2 billion people worldwide. To overcome the micronutrient malnutrition, many strategies such as the dietary diversification, supplementation, food fortification, and biofortification are being adopted. The main advantage of biofortification through the breeding approach is cost effectiveness and high sustainability. Pearl millet is a climate change resilient cereal which can grow on infertile and marginal soil under the limited soil moisture condition and high soil temperature where no other crop can grow. It is one of the most widely grown millet and provides the food, fuel and fodder in Asia and South Africa and covers more than 27 million hectares of cultivated area. Pearl millet is a good source of energy, carbohydrates, fat, ash, dietary fiber, iron, zinc and many other minerals. Due to the excellent nutritional property and resilience to climate change, pearl millet along with the other millets is renamed as a Nutri-Seed. The objective of my current study is to map future for grain, iron and zinc concentration in pearl millet using real population developed from biparental process. The present experiment was carried out with a mapping population of 210 F6 tribe 7 recombinant inbred lines to map the gene and genomic region for grain, iron and zinc content in pearl millet. Parent 1, that is PPMI 683, having a high grain, iron and zinc content, and parent 2, which is ppmi 627, has low grain iron and zinc content. Phenotypic evaluation of rail along with the checks were carried out at three diverse geographical locations representing all the three permanent growing agroclimatic zones of India, which is Zone A1, Zone A and Zone B. The evaluation was carried out for 3 consecutive years during the Kharif 2014 to Kharif 2016. Growth rates were approximately normally distributed when analyzed across the year at their respective locations. This is a pooled ANOVA of reels along with the parents. We show that ANOVA of 2 10 reels along with both parents revealed that genotypes taken in this study differed significantly for all the traits under study. A linkage map of 151 polymorphic markers were constructed by using MapMaker software. A total of 151 well-distributed markers between the ppmi 683 and 627 were used to construct a linkage map. Based on this data, the genetic map and the tutorial was constructed using the software ICI mapping. The linkage map had a total map length of 3234.4 centimorgan and the average interval was 8.69 centimorgan. In the present study, we attempt to identify QTLs for average mean for iron and zinc concentration in culminate grains. QTL analysis was conducted by composite interval mapping by using software QTL ICI mapping. This is a linkage map with QTL position of the permeate. Here red color is representing the zinc QTLs and blue color representing the iron QTL. The analysis indicated the presence of 4 significant common QTLs for the grain iron flanked by the following markers: ppmsb1, ppmsb2, IPS 0142, IPS 0180, third is IPS 0157, IPS 0093 and last is IPS 0206 and IPS 0015 on linkage group 2, 3, 5 and 7 respectively. For the zinc, one common QTL was found in E1, E2 and E3 location on linkage group 3. ANOVA revealed significant genetic difference in real for the grain, iron and zinc content. A wide range of variation was found in the present experiment material. Both traits are highly correlated. These are the ICAR markers which are associated with the grain, iron and zinc content. Marker trait association identified in this study provide a basis for further breeding program to enhance the level of iron and zinc content in culminate grain. This is the list of publications related to the present study. Thank you for your attention.
Thank you, Dr. Tripti, for such an engaging talk. Now we will proceed to the question and answer session. The participants in the auditorium who have questions can raise their hand and our volunteers will come to them with the mic. And online participants can type their questions in the chat box and our AV team will relay them to us.
I have a question for Menshchikov. Sorry if pronounced wrongly. As you have presented on hydropanels, I have some 1 or 2 questions. As we know, the water shortage is mainly in the arid and hot regions. Is your hydropanels are efficient to produce water in arid regions? And other one is, as you have told that for 1 meter square panel, you can produce 4 to 5 liter of water per day. May I know the cost of or cost for production of 1 liter per day? What is the cost for production of 1 liter per day? And have you applied your technology at commercial level? Thank you. Hello? It's not audible. Please unmute yourself.
Can you hear me?
Yeah, now. Loud and
Yeah,
clear.
okay,
Thank
thank
you.
you. Yeah, thank you very much. Thank you very much for your question. Actually, the cost of the 1 liter of water produced by our Hydropanel depends on the business model of the employing of our device. For example, if we consider for example, 2-3 years period of usage of our Hydropanel, the 1 liter of water will have a price which is equal to the price of bottled water. So it is a device, and it can produce water not only in. Of course, it can't produce water for example, it's not very efficient to use these panels for producing water in huge amounts. Currently, we speak only about the systems for drinking water. Of course, in the future, we can build farms of hydropanels for producing, for example, tons or thousands of liters of water. But now we speak for households and small businesses segments. And the cost of our hydropanel, of course, depends on its operation and depends on its capacity, and it varies from several hundred of dollars to thousands of dollars for one hydropanel. And so the final cost of water depends on the period of its usage, of course. If I understand your question right.
Thank you.
Thank you.
Hello. My question is to Dr. Chaomin from Hubei Academy of Agriculture Science. Is he there?
I'm here. Can you hear me?
Yeah, Dr. Chaomin. My question is, apart from the polysaccharides, did you look for any other metabolites, for example, secondary metabolites in your fungi or mushrooms?
Excuse me?
Yeah.
I haven't hear you clearly.
Yes. Apart from the polysaccharides that you are working on it, did you look for some other secondary metabolites? Or even did you go for some wild eatable mushrooms in your area in China?
Yeah, in China there are a lot of mushroom come from the wild, but in our study, I isolated the polysaccharides from the mushroom. These mushrooms are artificial cultivated mushrooms. Yeah.
Did you try for any other secondary metabolites? Did you have some methods?
I haven't determined these components.
Thank you so much. Yeah, thank you for your presentation.
Thank you.
Yeah.
I have one question to Ekshipova. Sorry for the mispronunciation. You have collected some work in Caspian region. So I'd like to know whether this whitest species wild population is available in Caspian region still now. Okay, I repeat the question. Actually, your work area is in that Caspian region where this whitest species origin is there. Whether the wild population is still available there.
Sorry.
This grapes Vitis vinifera species, no, so originating from Caspian region. Whether the population is still available?
There are pastures on which she is working. There are no species like. वहाँ जो जमीन है वो ऐसी स्पेसीज वगैरह कुछ पैदा नहीं करती है मैम.
Thank you all the participants for questions. Now, in the interest of time, we will move on to the thematic discussion.
So a huge round of applause for all the speakers, both online and offline, for their very nice presentations on this very theme. We heard from our speakers who were from different SEO countries about the research on this particular area of agriculture and food processing from various perspectives. And on this occasion, we would like to invite you all to a very interesting discussion on this theme where we can summarize the insights we gained from both sessions today, as well as have some insightful discussions about ongoing research in this area across various countries. We kindly request all our offline speakers here in this auditorium from today's sessions to take the stage for a thematic discussion. Now also let me introduce you to all the faculty chairs for this session.
Hello, we request all the participants the offline speakers to be present on the stage for thematic discussion, please. Meanwhile, we will introduce the speakers to you.
Now let me introduce now to all the faculty chairs for this session. Dr. Kesavan Subaharan completed his PhD from Tamil Nadu Agricultural University, India. Thereafter, he had worked on neurobiology of learning and memory of Drosophila melanogaster in National Center for Biological Sciences, TIFR, as DBT postdoctoral fellow. He had been a visiting scientist at Department of Evolutionary Neuroethology at Max Planck Institute of Chemical Ecology, Jena, Germany during 2008-9, and a visiting fellow at CPMU, JNCSR during 2011. He is currently serving as principal scientist in Division of Germplasm Conservation and Utilization at ICAR, NBAIR, Bangalore. His research interest is delivery of semiochemicals for pest management using the nanotechnological approaches. And our second faculty chair is Dr. V. Sridhar, who completed his PhD from IARI and thereafter joined ICAR IIH at Bangalore as a principal scientist. He is specialized in insecticide resistance management, climate change, and pests. Now I would like to invite Dr. Kesavan Subaharan and Dr. V. Sridhar to chair this thematic session.
A very good afternoon to you all. At the outset, I would like to thank the SEO, Department of Science and Technology, and this wonderful organization, Jawaharlal Nehru Center for Advanced Scientific Research, and in person, the president of this organization, Dr. Kulkarni sir, and the conveners, Ravi Manjutaya and his team, for giving me this wonderful opportunity to come and participate in this conclave and hear the works that is being done across the member countries. And it's wonderful to note that a lot of interesting works have been done. And just to quickly go into or kickstart this discussion, a quick recap of what has happened for the day is we have been assembled here for the agriculture and food processing session, the technical session 7 and 8, 7 presentation each, so totally accounting for 14. And it has touched upon all the disciplines of agriculture. Very interesting, right from the sensors for moisture requirement, the satellite imagery for moistures, or for the natural resource management. And most interestingly, as the world is going towards the green, a lot of interest has been shown by many researchers across the participating countries on trying to extract natural products and use them for the well-being of crops and for the wet animals also. A good sign the way in which the work has been progressing. Now, of course, a lot of concepts has been tried and the results have been spelt out, but this will not be in full unless if you people don't come out together and try to develop some concepts where you can have a dialogue. Now, one thing I would like to appreciate about this conclave is that it is not that agriculture people alone are going to sit and talk. You have a battery of scientists from artificial intelligence, disease and health, and other areas too. Pick up this opportunity and try to interact with them and try to club in the concepts that is applied in your area with their expertise and try to bring out a solution in toto rather than in isolation. And I have a few suggestions to kickstart the discussion based on the talks that I have been hearing. Wonderful talks have been delivered on sensors, right from Dr. Bacteriophages. And he has developed sensors to identify the moisture stress. But it's also interesting if they could start thinking of sensors for the biotic stress too. And if you have to try, I'm not just limiting to few of the scientists, I'm just, what has come to my mind, I'm throwing up for the discussion. Later on, you would have time to pick up the threads. If this team could integrate with Bapadas, who has been working on the satellite imagery and trying to identify subsoils moisture content, a sort of sensors from their side, sort of imagery from your side, and all try to get somebody to work on the biotic stress, not only for plants, but also animals in that area. A sort of a grand collaboration can come. And such collaborations will be very useful in the at the time when the India is moving towards the celebration of the millets. So when millets are a very hardy crop, less moisture is required. But if such technologies are introduced, you could bring in new innovation into this area. That is one aspect. Again, pardon me if I'm just restricting to a few examples because I'm just throwing open the discussion. Next, there is a gadget that was developed by one of the members from China talking about the quality assessing parameters, a very wonderful gadget that is very much required for the post-harvest. Now, that gadget is limited only to the nutritional indices. The bigger problem in the other groundnut growing area is an aflatoxin by Aspergillus, as rightly pointed by one of the queries that was raised during the presentation. So maybe if you can integrate with the team members here in trying to develop a gadget which address the aflatoxin also, then it is a value addition for the existing gadget. Now, very interesting information that I could gather today from our Uzbekistan friend, Dr. Shamshev, is that moving from potato to sweet potato, wonderful, because considering the climate resilience strategy that you are trying to adopt. But at this stage, I also would like you to look upon the various aspects where you can try to study the stress and also look into the transboundary pest that could happen, because now the movement of people and the commodities are very less. But there are certain very notorious insects on the sweet potato weevil, which is that should not cause a problem to you. As a proactive measure, you can discuss with the scientists over here and think if you can develop thing. Another interesting area is the extraction process of curcumin, and also a technique that has come out from our Chinese counterpart on maize, MACE, a very wonderful method where if you could integrate with your own team members from your country on trying to isolate polysaccharides from mushrooms, or if it could be employed with the curcumin extraction. Such sort of collaborations can bring in novel method of extraction of the actives. So with this small ideas which I'm throwing in for the discussion at this point of time, I also invite my chair if he has suggestions or any points to make.
Thank you, Dr. Subaharan. I'm totally agree with you here. When you see the overall presentations, the one means 4 or 5 presentations are revolving around this climate resilience, how best we have to come prepared, either it may be a crop replacement slowly or in terms of wherever this moisture stress is happening in terms as a abiotic stress. We need to get ready with the sensors and other things. One thing I would like to comment is as a group, is it possible to come out with the composite sensors? As of now, in majority places, only a single sensors are available. Suppose like along with the moisture sensors, can we add fertilizer sensors like nitrogen, phosphorus, potassium? These reductions, whether it is possible, that way you can look. And natural products, definitely it is going to be very important. Keeping in you the, whenever there is a glut of products quoted by the speaker, COVID as a situation, even if it is now assuming that COVID has gone in the other phases also, when the production is more, when the crop has a potential, what are the different ingredients, very important ingredients at lower cost, we can extract and we help the farmers. That is really going to be a useful thought in that. Apart from this, usage of omics platforms for coming out with resistance, that gives a flip and foolproof mechanism to come out with some of the resistance sources can be exploited. These are some of the observations. We'll participate in the discussion in due course. Thank you.
Sort of a quick rapid fire round, I would say, based on the knowledge that you have gained and based on what you had in your kitty. What is that you plan? You can just spell it out among yourselves, and maybe at a later part of the time, you all can even draft a concept note before you leave this place and submit to the organizers. That will go a long way. Thank you. I kickstart the program.
Thank you, Professor, for your suggestions and for your recommendations and giving a chance to attend here. I would like to say it's like cooperation with between SEO countries. As a researchers, I found many colleagues. For example, Dr. Discussing, and actually we did some conversation future our working like together. And also not just agricultural sector. And also I found some professor from artificial intelligence, Mr. Mutal, he's sitting in the auditorium, and we're going work like together, subject from agricultural yield side and using in artificial intelligence to make some techniques for farmers. In the future will be useful application to create some application for farmers. It means this kind of conclave will help us to make our ideas such you said before your speech, like when our research will be full, when we apply all our knowledge like together, not just agricultural field, but other like artificial intelligence and others, and even medicine. If we work together, our research, our knowledge will be full. And thank you for the organizers, such kind of event and conclave. Thank you very much.
Can I respond? Actually, as I am from the background of natural resource management, I will be talking more about precision agriculture or natural resource management. So as we all know that our population is increasing, at the same time our resources are limited, means water or land resources are limited and those are degrading. So definitely we need precision agriculture, means precision applications of maybe water, nutrients. So in that endeavor, we are using remote sensing technology and open source data. So many satellites are available from China, but those data are not in open domain. So we are mostly using NASA satellite data or European Space Agency. So if the data is coming from their sensors also in open domain, so there is a large gap. Suppose today the satellite is passing over this region, and it will again revisit the area after 5 or 10 days. So if multiple sensors are coming into this picture, so we can have daily images, daily imageries. So that will help us to devise these precision applications of any, suppose water or nutrient. So if some policy decisions can be framed among the SCO countries on data sharing. Actually, this data sharing has came, I think, yesterday also and day before yesterday also regarding various data. So if some policies can be framed in future regarding the data sharing, that will be a great, I think, achievement from this conclave.
So thank you. So I do agree with my colleague regarding the the comments. So I'll try to answer from my perspective. As you know, the my area is of post-harvest technology. So we in the Indians, not about 33% of the crops after harvesting are being lost as a post-harvest loss. Is a huge loss for the country. And because of that, the market value, when we sell in the market by the farmers, the market value is drastically going down. And the efforts that we are trying, by using the natural products, the curcumin as one of the main constituent, to enhance the shelf life. Our main target is to enhance the shelf life, mainly focusing to the highly perishable crops. The shelf life of having the 7 to 8 days, maybe minimum 10 days, like that. So our target is to increase the shelf life so that the freshness, without hampering the biochemical parameters aspect in the fruits and vegetables, and so that it can reach to the general public as a mass. So I do agree with the chairman's of sessions. Yeah, we require a pool of the ideas for collaborations with some other the ideas, like the polysaccharide. As already the polysaccharides are being also utilized for the edible coating. It is there in the literature. So not only that, the another main problem is the post-harvest diseases. So this is also one of the area where we should be working together by taking the advanced technology and that already from the scientists from China. So they are also working. So we can come together having a a particular solutions for this particular aspect. Thank you.
My main area of work is fruit breeding, so I'll be talking a little related to that. We are always searching for the germplasm, always the resistant source. uh, Whenever we are working for the export crop or something, it's not indigenous to India. We're looking for some other countries. Mainly, in case of mine, the grapes, I'll be looking for China or Russian countries, and this pomegranate, Kazakhstan, and all. But we won't get any information from these countries. So, if there is any data, germplasm data repository for SEO countries, where this germplasm inventory is available, and their traits also, specific traits also available, it's very useful for us. And if any mechanism is required for this sharing of this germplasm, it will be very beneficial for us. And India can also share many indigenous crops like mango and all. We have germplasm availability. Such repository is available, it will be very useful for us. Same like that, genomic data availability also for crop wise, if it is commonly available, some domain or something for SEO countries is available, it will be very beneficial for the countries to work together. And also, so was talking about the sensors. Not only we can restrict to the soil or some structures, even for early screening of the disease diagnostics also we can introduce the sensors for image analysis along with the sensors, it will be very useful because for screening also, because especially in case of the perennial crops, we have to wait for many decades to develop a variety. To reduce that one, we are talking about the speed breeding or smart breeding and all. We can integrate all those things because I think many countries have all those techniques, especially I think China is having the very well robotics. They are very strong in that. So we can collaborate with that also. It will be very useful because labor cost is increasing day by day. So robotics in many harvesting or any cultural practices in agriculture, it will be very useful. These are my some of the.
Thank you, Indian professors. 1 minute.
He's saying that we want to use our ideas on Indian soil.
We are glad to cooperate and are ready to cooperate in solving common tasks. Thank you.
Уважаемые присутствующие, хотел поблагодарить организаторов за приглашение и участие в такой большом мероприятии. Это уникальная возможность поделиться своей работой, ознакомиться с другими работами и решать такие важные проблемы комплексно. И спасибо вам большое за это.
Highly respected members of SCO, we would like to thank you for your invitation to call us. We can cooperate with each other very well and we can work together. Thank you.
Now, thanks to everyone for being a part of this intellectual, very interesting and informative discussion. On this occasion, I'd like to present our faculty chairs with a small token of our gratitude. Thank you. With this, we have come to an end of this technical session. We thank all the speakers, chairs of the session, all the participants, volunteers, and our AV team, Srinath, Dilip Kumar, Munegovda, Mohammad Peer, and Srinivas S. Before we break for lunch, we have some announcements.
I hope you have been enjoying the session so far, and also the food that comes along with it. And to get you a taste of really how food is eaten in the southern part of India. So India is a very diverse country. If you go maybe 200 kilometers in any direction, the language changes, the cuisine changes, people speak, tradition changes. So there is a very peculiar and a particular way of eating in southern India, which is the south part where we are now today in Bangalore, Karnataka and all. We, instead of eating on plates, we have a banana leaf. And you'll be food served in banana leaf. And the correct way of eating is using your hands and not spoons and all. Okay, so Dilip is going to put a picture here. This is how it's going to be, okay? And unlike your round tables, you'll be sitting like this, and in front of you, each one of you will have a banana leaf, and the food is going to be served to you. You don't have to go and get the food. One by one, the food will come. You can use your hands. They will tell you how to eat the food. The idea is to take each curry, mix it with the rice, and eat the food. If you still don't like it, some people are like, don't want to use their hands, you can still use your spoons, but the food is more tasty when you use your hands, okay? So that's the first thing today. Okay, after the food, you also will get to see a rural village, how it is, like how is a typical village in India. We don't have to go so far. We are lucky that across the road, there is an institute called Mahatma Gandhi Center. And so if you go there, we have a tour for you, how a typical village looks, what people do in a village and things like that. So you'll get a chance of going and look at it. Our volunteers will also accompany you. Some of the people from India may feel not so very happy, but I have been assured that it's a very nice place. Everybody feels happy when you go to such a place. I hope you'll like that. After that, we have a cultural event. For some of you, I must have already told, the cultural event is a assembly of multiple different dance forms across all over India. So you get to see all the different dance forms. And after that, there is high tea. And my volunteers again will help you to come to the high tea place, the same place where we have lunch every day. And the artist will also join us. So you'll get to hear from the artist about the dance forms. And if you want to take pictures with them with their colorful outfits, you're more than welcome. And after that, we will be going for a conference dinner. Of course, it's open to everyone. And the buses will take you to the conference dinner place. It's an open lawn area with some lights. I hope you will enjoy that evening. And after that, you will be taken back to JNCSR here or to your place in the hotel. Okay. So at any point of time, if you have any contacts, don't hesitate to contact me or through the volunteers if you have any issues. And I hope you enjoy the rest of your day. Okay. Thank you very much. So now we can start walking towards the dinner.